Welcome back to Science in Pajamas. Today in our biology class, we're going to be doing a Yeast Anaerobic Respiration Fermentation Lab. And I wanted to go through and perform it for any kid who is absent due to sickness, quarantine, etc. This way they can still see the setup and still answer the questions, the analysis, etc. So we're going to go through it, and we'll be able to see how it goes. So I have all of our supplies. We have our test tubes and our homemade, good old DIY large test tube holders. We've labeled all of our test tubes as tube 1 with 0 sugar, test tube 2 with 1/4 tablespoon sugar, test tube 3 with 1/2 tablespoon sugar, and test tube 4 with 1 tablespoon sugar. Notice that these have caps. I'm going to set them up here so they can be nice and seen when we look at them later. I have a lot of sugar because we haven't done the lab yet, so I got just a lot of sugar, so we have it. I have some water already warmed up. It does not have to be hot. It just has to be warmed up. You can put in the microwave for about a minute. I have a hot plate here that I just have it kind of warming up on. I have some yeast that I picked up, and I have some balloons as well. Now at school, and by the way, sorry about my throat. I have a sore throat over the weekend, so bear with me. I don't have any tablespoons at school. Now, in your kitchen, most people have, or many people have, 2 kinds of spoons. They usually have like a larger dinner spoon and a smaller dessert spoon. The smaller spoon is more or less kind of the equivalent of a teaspoon. The larger dinner spoon is the equivalent of a tablespoon. Most plastic spoons are the size of the smaller dessert spoon. 3 of these would be about the size of a tablespoon, so give or take. Now, this doesn't have to be exactly precise. Ideally it should, but as long as we kind of keep it within the range, it should be fine. All right, so we have everything together. What we're going to start by doing is we already have our test tubes labeled. We're going to put about 30 milliliters of water into our test tubes. So I'm going to take our test tubes. We've already warmed up the water. I'm going to take the lids off. And the nice thing about the test tubes, they're labeled using the Erlenmeyer flask, so it can pour nicely to 30 milliliters. Okay, there's 30. Again, we want it warm but not hot. If you're having trouble holding the flask, then it's too hot for you. It should feel nice in your hands. If it's too hot for you, it's going to be too hot for the yeast. Okay, this lied to me. It said I had over 100 milliliters in here. So we're going to improvise a little bit. I'm going to get these so that they're all starting at the same amount. There's going to be about 27, 28. All right, so now we have that. Add sugar to the tubes in the amounts listed. All right, so tube 1 has none. Tube 2 has 1/4 tablespoon. Now, since this is, or 3 of these is 1 tablespoon, 75%, so 0.75 of this would be 1/4. Let me see. So, let me see. No, wait a minute. So I want 1/4. Okay, so I want to divide this by 4. So 4 divided by 3, 1/3. Oh man, this is that time of year. It's the week before break. So 3 divided by 4 is 0.75. So yeah, 1/4 would be 0.75 because, yeah, 0.75. All right. So yeah, I was right the first time. So I only want about 1/4 of a teaspoon in here to be okay. Of a tablespoon. Shake it up to mix it. Shake, 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 because we want to dissolve the sugar in. I know there's none in there, but I don't want any sugar to fall in, so I'm going to just cover that up. Make sure that's all dissolved. This is half, so we want 1 full scoop. One full scoop and a 1/2 scoop. And then we're going to put the lid on and we're going to scoop, shake it. Shake, 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 shake. Mix it up. Dissolve that sugar in. And then this one's a full tablespoon, so we're going to want 3 full scoops. 1, 2, and 3. Okay. Like I said, ideally it should be accurate or precise. If it's not, at least the point is to get none, some, more, most. All right. Let's see, we're going to add 1/4 tablespoon of yeast to all of the thingies, all the test tubes. And then we're going to shake to mix the yeast in. One moment. All the motion centralized. There we go. All right. God bless it all. Okay. So the point is we want about the same amount of yeast in all of them. See, they're living things, so we want to make sure that we start off with the same amount of life. This way we can see if starting off with the same amount of living things, will yield the same amount of respiration. Because if the same amount of living things yield the same amount of respiration, then that means that we have the same amount of life processes. But if we see more life going on in some, more respiration going on in some, that means more life processes is going on in some. Mix the life forms in. We'll talk more about what's going on after I mix these together. Okay. And now the point of the balloons is to collect any gas that's produced. Oops, that's not good. Okay, so we got one. We've got two. Let's get that balloon on. By adding the balloons, like we said, it's going to collect any gas. This will create a closed environment so that we can look for any foam, gas, etc. Now, the reason why we're doing this, I'm going to put these up here for now. We've been talking about respiration and anaerobic respiration. So let me get my timer started because we have to take a look at these different intervals. So, Come on. There we go. All right, so what is respiration? So a lot of times we think about respiration as what happens when we breathe. We breathe in oxygen, we breathe out carbon dioxide. But the question is, well, why do we do that? That's respiration, that's the respiratory system, but what's the point of it? That's actually a very easy question. Respiration doesn't just refer to breathing, it refers to a process of making energy inside of our cells. That's what cell respiration, cellular respiration is. It's our cells creating energy. And our cells create energy in order to power our cells. Now, most kids know what the mitochondria is. Again, I apologize for my voice, but the mitochondria is what most kids would call the powerhouse of the cell. In other words, it's the organelle that powers the cell. It takes glucose, which is a carbohydrate. It's a simple carbohydrate, and it uses it to make energy. Just like how we need energy to do stuff, or our phones need energy to do stuff, our cells need energy to do stuff. So our cells get energy from glucose. That's why we get tired when we don't have food, why we get or feel like we don't have energy, because our cells get tired. So we get tired, and when we feel like we don't have energy when we don't eat, and that's because our cells don't have energy. So what happens is we eat, we digest, we get glucose. The glucose gets sent from our digestive system through our bloodstream and to our cells, where the glucose is then sent to our mitochondria. Once in our mitochondria, the mitochondria will break the glucose, releasing the energy from the chemical bonds and storing it in a molecule called ATP, adenosine triphosphate. And it'll use that to then essentially think of ATP like a rechargeable battery. The ATP will go through and power every single thing that goes on in the cell. Now, that's just part of the story. The thing is, the mitochondria can't do this on its own. It needs oxygen in order for this process to occur. So that's why we are inhaling oxygen, why we have a respiratory system. We inhale oxygen. Oxygen goes to our lungs. The oxygen goes from our lungs to our bloodstream, from our bloodstream to our cells, and from our cells to our mitochondria. And it helps our mitochondria to break apart the glucose in order to create energy. One of the waste products of this process is carbon dioxide. And we get rid of that carbon dioxide waste when we exhale. So that's what cell respiration is. It is a process of us using oxygen to help our mitochondria make energy from glucose. Now, what does it have to do with yeast? Because they're not using oxygen right now. Well, what's going on right now is this cell respiration is a form of energy, or a form of process to make energy called, I've rubbed off some of the things. I apologize. I'm just adjusting the balloons, making sure they're on all the way. But I digress. Anyways, I apologize. Whew. That one shot up. Got some weird results going on. I apologize. Anyways. Interesting. Sorry. We're about to reach the end of the first time, so I'm going to pause right here. All right. So what we're going to do now is I'm going to hold each one up to the screen or to the thing. It says in the directions you are to calculate the volume of the foam by finding the measurement of the height of the foam in millimeters, as well as the volume of the tubes, or by finding the volume of the, anyways. First, you also have to collect the, you have to take pictures of the tubes. So if you want to take your screenshot, here is for 5 minutes. I'll let you take your screenshot for your picture. And then, for one at a time for the images. Wonder if I mislabeled these. I must have mislabeled them. Because you can see the foam all the way down there. Yeah, I mislabeled these. Okay. Yep. I did mislabel these. So the foam you can see starts all the way down there. So the foam starts all the way at the bottom there. So you can see how it's how it's not just water. I think I mislabeled. You can go back in the video and rewatch. But definitely go back in the video, rewatch, see what's going on. But there's that one. I'm just going to hold these up. You can rewatch as needed. See, this one's showing better foam. You can definitely see the foam up there. So I'm going to show that one. You can even see the foam all the way up in the balloon. That is really impressive. So from the bottom all the way up into the balloon, that's some impressive foam. Pull that down a little bit, make sure it doesn't shoot up. I'm going to go back to number 1 for a moment because foam should just technically be that bit right there. But I don't know. Getting some weird results, so I'm not quite sure what's going on here. This is supposed to be number 3. This one looks pretty good. So this should be tube 3. This is supposed to be tube 4, but I think I got tube 1 and tube 4 mixed up. Hold on a minute. But there's not a whole lot going on, so I'm not quite sure. I'll have to see what goes on with tube 4. Okay. I'm going to let those sit for another 5 minutes. For our 10 minutes, and we'll see what goes on. All right. So we got our 5-minute data. Now, to kind of go along with that, hopefully you were able to pause the video, get your measurements for the heights. It says you also need the radius of the tubes. So that should help you get for the radius. So that can help you get the radius for that. All right. As you're aware, I don't give you guys the answers, but I provide you with the tools. So I showed you what the diameter of the tube is. You can find the radius. I held up the tubes so you can get the screenshot. I held up each tube individually with the ruler so you can get the measurements for the height. So now that you have the measurement for the height, the radius, you can calculate the volume. All right, now where were we talking? Oh yeah, the yeast. So the yeast are doing their own separate thing. The yeast are doing what's called anaerobic respiration. So cell respiration, what our mitochondria does, is aerobic respiration. That means that it uses oxygen to create energy. But the yeast are doing anaerobic respiration, which means that they are using no oxygen to create energy. Now, specifically, they're using something called alcoholic fermentation. Now, we should be familiar with alcoholic fermentation because we actually use it all the time. And no, I'm not just talking about to make alcohol, because yes, humans have been using that process for a long time. Beer, wine, etc. But this is also the process that we use to make bread. That's why we use yeast, to make bread. And that's what is going on here. Now, what you might not realize is, besides alcohol, one of the byproducts of alcoholic fermentation is carbon dioxide. Hence, the gas that's being produced. That's why when when a baker is making dough, why the dough rises, why they let it sit. They add the warm water and the sugar and the flour and the yeast. The yeast wakes up, it's dormant, and the warm water wakes it up. And then the sugar is a food source for it. After they're eating the food, they're going through respiration, they're going through fermentation, they're just doing normal life processes. As they're going through their normal life processes, they're just, doing their normal thing. They're hanging out, doing their normal life thing. But in the process, they are creating carbon dioxide. They're exhaling, just like we do. And that exhaling is causing the bread to rise. That's why oftentimes the baker will beat down the bread, and then they'll let it rise again, and then they'll beat it down. Now, the reason why they want to do that is because as the yeast is going through, the other byproduct, the ethanol, does help to give the bread some flavor. Now, when you bite into bread after it's been cooked, that's why you see all those pores in there. Those are the air pockets from the carbon dioxide. Now, you don't taste the alcohol at that point because it's been cooked off in the baking process, so you don't get drunk off of bread. But that is what's going on in this idea, in this process. So that is what is going on here. This is what we are witnessing. This is alcoholic fermentation. This is anaerobic fermentation. This is what yeast do. They create energy just like us. All right. So once again, you need your screenshot. So let me get out of the way. Yeah, there's your picture. Let me see it in the screen. Oh, there we go. So take your picture. It does kind of smell like bread right now, by the way. All right, so tube 1. Remember, hopefully you're remembering the directions say measure in millimeters. Got a little bit of foam there. It's kind of cloudy all the way down, so I'm not quite sure if all that is foam. It's really hard to tell. There's no gas in the balloon, but it's foamy all the way down. But definite layer of foam at the top. Number 2, big old bit of foam all the way into the balloon. Big old layer of foam all the way into the balloon. You can even see it in there. And remember, you can pause these videos so you can get your measurements. Not as much going into the balloon, but you can definitely see the foam in the tube. And that's another reason why we have the balloons on top. Just kind of catch them in case the foam goes anywhere else, like with tube number 2. Not really much going on with tube number 4. I'm not quite sure what's going on. Maybe the water wasn't warm enough. But you know what? That's why we do these experiments. Or maybe I did switch 1 and 4. You can always go back in my video and double check. Or maybe there's just too much sugar for it. Not quite sure. All right. We gotta do one more 5-minute interval. All right. I know this is a long video. We've stuck it out this long. Might as well stick it out the rest of the time. Now, yeast, if you're wondering what they look like, they're just really itty-bitty little things. I mean, you can kind of see them. Oops. They're just little microorganisms. If I remember correctly, they're more like a fungus. Let me just... Saccharomyces cerevisiae. Yep, it's a fungus. The fungus among us. It's amazing, though, if you really think about it. We always used to think fungus is so dirty and bad. that fungus is so bad for us. But now look at us. I mean, we use fungus to make bread and alcohol and things like that. We use fungus to make blue cheese. We use fungus to make antibiotics, penicillin. So, they're not as bad as we like to think they are. Yeah, I know, I'm just kind of thinking. But, just kind of cool how things work out like that, if you really, really think about it. Science has actually been pretty cool. And things aren't always what they seem. And take these little guys. Didn't really think they were much to look at, much to do. Bread's pretty darn tasty. I know people always give carbs a bad reputation because, carbs are bad for you, carbs, blah, blah, blah. There's no such thing as a carb-free diet. You can reduce the number of carbs you eat, but there's no such thing as a carb-free diet because you need glucose in your diet. Otherwise, your cells would die and thus you would die. So just keep that in mind. You can reduce your carbs. You can reduce your starch intake. You cannot get rid of all carbs. But yeah, so this is our lab. So what you need to do, since we have 2.5 minutes left before we take our final data, in the lab, and I have it open on my screen, which is why I'm reminding you, you have to calculate the volume of foam. I've been showing you the data. Go back as needed. To find the volume, it is the height of the foam times pi times radius squared. So radius is the diameter of the tube, the opening of the tube, divided by 2. A little while back, I took another tube. I showed you the opening of it with the ruler stretched across it. I've been showing you the height at each 5-minute intervals. So make sure you collect your data so that you can find the volume each time. Let's see. So you're finding the volume at each time interval. Let's see. Yep. Okay. Just making sure I'm getting everything. Good. You'll get your last picture in a moment, and then you'll be able to explain what's going on. I guess the last thing to really discuss is then, where does this occur? Now, we mentioned that cell respiration occurs in the mitochondria. That's not where fermentation occurs, because cells that do fermentation, like bacteria cells, they don't have a mitochondria. So this process, this just takes place right in good old good old cytoplasm. It just takes place in the cytoplasm, nowhere else. No special organelle, nothing. The glucose that's taken in by these cells just gets broken down right then and there in the cytoplasm. No special organelle, and that's why fermentation doesn't yield as much energy. Fermentation and anaerobic respiration, it doesn't give off as much energy because it's when you're a smaller, less complicated organism like a bacteria compared to a multicellular eukaryotic organism, you don't need as much energy to keep the cell going. So you don't need to make as much energy. So this does not make as much energy. All right, so last picture for the assignment. Go ahead and take it. Did you get it? Oh, sorry, there you go. All right. And now your last measurements. This is T1. For T1, foam. There's a definite foam. That's the potential foam. Again, I'm not quite sure. I definitely see foam up here. It's all kind of weird colored all the way down though. Okay. Now here's the interesting thing. Look, Foam is starting to disappear. The foam is coming down. The foam is going away. Huh. Imagine that. The foam is no longer all the way up at the top. The foam is only about... There is still some, I know it's hard to see, some bubbles being produced. So there is still some of the reaction going on, but there's not as much. So not as much of the reaction, not as much of volume. Okay. Get your data for number 3. Sorry. And then data for number 4. Alrighty. I know this was a longer video because we had to wait those 15 minutes, but I hope this helped, and I will get this taken care of as soon as possible. Alright, you guys, I hope this video was beneficial and it helped you guys out and helped you understand anaerobic respiration and fermentation. Otherwise, until next time, stay safe, stay healthy, and take care. Bye-bye.